Hello everyone, we are solving question number 12 from GATE 2017. It says enzyme-linked immunosorbent assay ELISA is used for A. Quantify antibody level in blood, B. Determining the molecular weight of an antigen, C. Purifying protein from biological fluids, or D. Determining the molecular weight of an antibody. So ELISA is an immunological assay which is commonly used to measure antibody, antigen, protein or glycoprotein in a given biological sample. It is to measure the quantity or the amount of these molecules that are present in any of the given sample. So there are many types of ELISA, mainly 4 types. In direct ELISA, you have your antigen that is present in your sample. So you use antigen which you want to detect antibody against that antigen, and that antibody is bound with a enzyme, and once the primary antibody which you use to detect the antigen bind to the specific antigen in the sample, you provide a substrate, and this enzyme which is conjugated with the antibody generate a response either in a form of color or in the form of light, and that can be detected. In indirect ELISA, you have antigen, you use primary antibody against that antigen, and then you add secondary antibody that is conjugated with the substrate, and then you use a particular substrate, and that particular substrate generator signal which can be detected. Then you have sandwich ELISA. In your sample, you have a particular antibody and you want to measure the antibody, and then you use antigen against that antibody, and this antigen is bound to the antibody that are present in the sample, and then you use another antibody that you can bind to the same antigen, and this primary antibody is followed by the secondary antibody which is conjugated with the enzyme, and you provide a substrate and it generates a color. So that is sandwich ELISA. And when you have a competitive ELISA, then you have an antigen in your sample, and then you use antibody that is bound to that particular sample, and that is associated with a particular enzyme giving a color, and you add another inhibitor antigen which inhibit the interaction of this particular antibody with the antigen, and this replace the this particular antibody first antibody, and there is a reduction of the color that is generated. So if there is a competition, then you will find the reduction in the color, and that's how you see the competitive assay here. So and this is again the same thing. Then you have, there is a competition, then there is a sandwich, and then there is a direct, and these are the enzymes that are generally used as a which are conjugated with the antibody, be it primary or secondary, and you use their respective substrate and they change color, and you take the you stop the reaction and you take the reading at this particular nanometer. So coming back to the question, so enzyme linked immunosorbent assay is used for quantifying the antibody level in the blood cell. Yes, it is used for determining the molecular weight of antigen. No, this is wrong. And it is for purifying the protein from biological fluids. No, this is wrong too. Or determining the molecular weight of the antibody. No, it is not you helping to determine the molecular weight of either antibody or antigen. It is just used to quantify the antibody level in blood. So answer here is A.